minute, invisible to the naked human eye. It takes the all-seeing scrutinizing power of a scanning electron microscope to force this silent killer to reveal itself, a sphere with a distinctive halo of spikes.
拍到世界上第一张的电竞照片，病毒呈现了典型的冠状病毒的形态。
我们第一步的工作就是要开展这个样本的一个检测跟病毒分离，我们才有可能验证这个病源。
能看到这个颗粒，这说明我们的病毒分离是成功的，
才能开展后面的疫苗和药物的研发。
It is culprit number one in a pandemic that, within less than 2 years, has infected over 200 million people and cost the lives of more than 5 million, SARS-CoV-2. 2019, news reports began to tell of cases of a mysterious new form of pneumonia. The coronavirus was back. Soon, the hope of the world would be a medical breakthrough first made 200 years ago. Vaccination. The story of coronaviruses goes back to June 1964, when Scottish virologist June Almeida tested a nasal sample obtained in London 4 years earlier. Examining the sample through the electron microscope, her attention was drawn to something unusual. Each of the grey dots before her was surrounded with a halo of even smaller dots. She'd discovered a new type of virus. It was this conspicuous fringe of dots that earned this family of viruses its name, because this halo made these viruses look as though they were wearing crowns. Corona in Latin. Scientists took to calling them coronaviruses. In the early 21st century, coronaviruses were responsible for 2 lethal outbreaks, SARS and MERS. But it wasn't until 2019 and the emergence of COVID-19 that a coronavirus began a rampage across the world that would transform it into the common enemy of all mankind.
自从人类文明产生以来，一直面临着瘟疫的威胁持续存在。人类的这个历史文明的进程始终受到了传染病的威胁。
人类认知病原微生物也就是100年来，过去我们都不知道它里边是什么，是传染病是由病原引起的。
Our world is home to a host of invisible microbes capable of causing disease: bacteria, viruses, protozoa, and fungi. In large measure, pathogens such as bacteria and fungi have been conquered by developments in antibiotics and other medicines. But viruses pose special challenges. Lacking an independent cell structure, they can only survive in a host. For that reason, the surest line of defense against viruses lies in bolstering the human immune system. The first ever vaccine was developed in 1796, when the British physician Edward Jenner developed a method of preventing smallpox by exposing people to the smallpox virus's relatively harmless cousin, cowpox. Long before vaccine, Chinese doctors used an inoculation technique in which smallpox scabs were ground into powder, packed into a pipe, and then blown up a person's nostril.
面对这样极狡猾、极凶险的这么一个新冠病毒，所以基于人类过去成功战胜病原微生物的这个经验，我们要做疫苗。
In the first half of 2021, the COVID-19 vaccination began to be rolled out. Slowly but surely, the domestic outbreak of the disease was brought under control, and life returned to the cities. The responsibility for developing COVID-19 vaccines has largely fallen on shoulders of this ordinary-looking man. He's happy with the progress made in this ambitious project, but is all too aware that many challenges lie ahead. For Tan Wenjie, there can be no let-up, no room for complacency.
对，现在疫苗 接种的人群血清的那个中和实验，现在免疫的这个血清，疫苗免疫的血清这变异株没有中和。
The IVDC, China CDC, short for National Institute for Viral Disease Control and Prevention, Chinese Center for Disease Control and Prevention, is on the front line of global efforts to develop COVID-19 vaccines. One issue above all gives the scientists at IVDC sleepless nights: the emergence of an increasing number of coronavirus variants.
经理，你这个分析什么序列？
就是之前做的一个血样本，还有。
Thanks to the national COVID-19 surveillance system, Tan is able to keep the coronavirus under constant scrutiny, providing invaluable data for the vaccine development initiative.
这个93.53是他测出来的。
对，现在能分出来的它的所有病原的一个占比。
The IVDC-CDC has been at the forefront of the fight against the virus, tirelessly doing its bit to push forward vaccine research and development.
我们现在对新的传染病都易感，但是我打了疫苗了，我产生了综合抗体了，实际上对易感者，对我们实际上对人，我有保护了。
The story of this remarkable fight against disease began in the closing days of 2019, when hospitals in Wuhan were left scratching their heads by cases of a mysterious respiratory illness, a pneumonia of unknown etiology. At the time, the only clues were a common set of symptoms and similar chest X-ray profiles. What was causing it? The only way to tell was to sequence nucleic acid from specimens taken from patients.
所以31号一早呢，我们就有专家派到前方去，病毒病预防控制所。这个所的最重要的任务就是不明原因的病毒病的应急的处理。
Soon, a sample was winging its way from Wuhan to the IVDC. So little was known about this pneumonia of unknown etiology at this point that there remained doubt that it was caused by a virus at all, let alone COVID-19. But one thing was sure: combating the disease successfully would depend on identifying the precise pathogen.
我们需要这些活的东西来做研究，毒种是我们进行科研的一个最基本的一个支撑。
As dawn broke on January 2nd, 2020, the tension was palpable at the IVDC building at CDC. Identifying the culprit behind this pneumonia of unknown etiology in the shortest possible time was literally vital. For the scientists, it was a race against time. The moment the sample was received, Wu Guijun, the CDC's chief expert in biosafety, leapt into action, organizing the isolation and sequencing of the sample. Only by isolating and culturing the virus could the scientists take the first step on the road to vaccine development. But this is never an easy task. In order to do so, a virus must be successfully isolated from a tissue sample, then cultured in a pristine environment. Only when that's done can the really hard work of vaccine development begin.
为了第一时间生产出灭活疫苗，我们是在顶着压力、顶着风险，然后也是在科学验证的情况下，这么来开展的这个工作。
But circumstances made the task of virus isolation even more challenging than usual. Dealing with an unknown pathogenic agent, the scientists had to work under the most stringent safety conditions in the P3 lab, Biosafety Level 3. Working in conditions of negative pressure to ensure that nothing leaked from the lab challenged the physiology of even the most experienced workers in the field.
在 P3 实验室，它那里边一般来讲，压力是-50%到-60%， 所以在里边呢人会不舒服。
Worse, scientific researchers working at the P3 lab had to wear a hermetically sealed protective suit and carry a rebreather. Denied access to the free circulation of air, working under unimaginable pressure, the temperatures these dedicated workers had to endure for hour upon hour are hard to imagine.
从那天开始呢，实际上已经进入了24小时在那工作的状态。我们病毒病所新的这 P3，就7个，几十种高致病的病毒，在这里是可以操作的。所以呢，只要发生不明原因的，我们这地方是可以进行的。
Isolating a virus involves introducing a specimen containing viruses into a suitable cell. Virus isolation has been successfully achieved once viruses in the cell start to multiply and become culturable.
那个时候就真的是在跟病毒赛跑，这回一点都不夸张。
晚上大约是11点多的时候，然后我们一看，确实是有序列，而且一比就是一个新的冠状病毒。
On January the 10th, the breakthrough came. Chinese scientists took the lead in announcing the full genome sequence of the novel coronavirus and its SEM image in GISAID. Later, the WHO named the infectious disease caused by the novel coronavirus COVID-19, while CSG called the pathogen SARS-CoV-2. Just 1 week after the arrival of the first specimen, the virus had been isolated and its full genome sequenced. Now the path was open to explaining the origins of the virus, its transmission, and how it might mutate. More importantly, this landmark would greatly speed up the research and development of detection reagents, vaccines, and antiviral drugs. At last, a faint glimmer of hope.
这种共享就为各国，就说这个研究包括预防控制赢得了时间。
Thanks to China's sharing of the full genome sequence of SARS-CoV-2 with the rest of the world, the United States National Institutes of Health could begin to work on the research and development of its first mRNA vaccine. As the epidemic gained ground across the world and struck with increasing ferocity, the global scientific community forged a united front against the virus.
国家卫健委昨天下午通报，
新型冠状病毒感染的肺炎确诊病例290，其中湖北72例，上海2例，北京3例，
浙江16例，江西2例，海南1例，贵州1例，宁夏1例。
But it was early days. On January the 20th, 2020, with the COVID-19 situation in Wuhan critical, the Joint Prevention and Control Mechanism of the State Council met to discuss full-scale anti-epidemic measures.
现在可以这么说，是肯定的有人传人的现象。
With vaccine development still in its infancy, one method alone could take the battle to the novel coronavirus and check its speed: strict quarantine measures.
中央决定封城。
The Wuhan lockdown was a stark reminder of the urgency of the need for a vaccine. Led by the Joint Prevention and Control Mechanism of the State Council, vaccine research teams leapt into action following 5 separate R&D routes formulated by the Ministry of Science and Technology.
第一条灭活，第二条蛋白，第三条腺病毒为载体的疫苗，第四条流感病毒为载体的疫苗，最后一条就是 mRNA。
Of all available types of vaccines, inactivated ones have the longest history. They date back to 1885, when French microbiologist Louis Pasteur saved a boy bitten by a rabid dog with a vaccine he'd created. The first attenuated vaccine in human history was born, sparking a medical revolution. By the 1990s, another type of vaccine had emerged, the nucleic acid vaccine. But there were 2 big question marks hanging over the viability of developing this type of vaccine. They took a longer time to develop and were more difficult to store and transport. And with SARS-CoV-2 spreading like wildfire, time was of the essence. Priority had to be given to the tried and tested technology of inactivated vaccines.
现在的这个疫苗研制，我们在考虑它安全性、有效性的同时，我们还在充分考虑它的可及性和可担负性。
我们实际上对灭活疫苗是有一定的信心的，总有一条大路是会走得最快、走得最通的。今天细胞怎么样？我们的任务还挺重。
现在这细胞主要是用来做检定，然后做综合抗体用。
But developing an inactivated vaccine would be no quick fix.
现在细胞需求量比较大了。
对，现在就一做综合实验，几百块卵子的量。
First off, scientists need to select and culture the strain of virus suitable for the creation of a vaccine. After that, they will need to be able to continuously subculture that virus. Only when a stable viral subculture is available could the progress be made on the development of a vaccine. After selecting and isolating a strain of virus to act as the seed for the culture, the next vital step is to get the strain to grow normally in a cell.
病毒它是一种寄生的这个生物，它必须依赖于一个宿主才能活。现在疫苗生产用的细胞 virus 细胞就是一个非洲绿猴的一个肾细胞它需要在这个细胞里边才能够复制，才能够生长。
把所有的细胞养起来，等着材料来开始分离，因为我们的判断说这个病毒可能很难分离。这是一个灭活灭抢了2-3个月的时间。
Then came a breakthrough, the discovery that SARS-CoV-2 grew in the clean cells. Could a COVID-19 vaccine be just around the corner? By January 19th, 2020, Wang Hui had long been working on plans to produce a COVID-19 vaccine at the Beijing Institute of Biological Products Company Limited. With interest in vaccines surging as a result of the mass COVID-19 outbreak, a seemingly outrageous thought occurred to Wang Hui.
那你的人也要跟进来把这个整体。做了一辈子疫苗，我还真没有经历过这么纠结的和挑战的一个局面。
Working under huge pressure, Wang Hui suggested that Yang Xiaoming, chairman of the board at China National Biotech Group Company Limited, should upgrade the current facilities so they could meet the standards required for COVID-19 vaccine production. To begin with, this was an eye-raising suggestion.
你不能等到疫苗成功，你再建车间，时间上显然是来不及了。
当时生物安全的这实验室，我们国家还是有的，但是高等级生物安全的能用疫苗的这个生产车间，我们国家就没有。
我2天了，你想的咋样了？他说是再考虑一下。
这个要修不修是要下决心的。第一，技术你能不能修，能不能按时修出来，这都是属于挑战性的。
然后呢，我24小时我又找他，他说拆吧。
换了毒株之后，这个原来的6点，现在6.5可能不是6.5了。我觉得可以参照蛋白原来的，我们不是做的248吗？
厂家的铝佐基做了所有的对比，我们选定了一家，就觉得排了个1、2、3的顺序，对吧？这些参数都已经完成汇总了。
On the Chinese New Year 2020, Wang Hui called on key workers at the Beijing Institute of Biological Products to discuss ways forward in the effort to develop the inactivated COVID-19 vaccine. Once Tan Wenjie's team from the CDC had completed virus isolation, her institute would have to take responsibility for subsequent production processes, including cell culturing and inactivation. In order to transition smoothly from vaccine R&D to production, Wang Hui immediately went to Tan Wenjie to learn about the isolation condition of the SARS-CoV-2 that could be used for vaccine development.
我们就奔赴了这个 CDC 病毒所，因为它需要 P3的条件，我们跟 CDC 的技术人员一起进行了前期毒种的这个制备和毒种库的建立。今天晚上你说结果是3点钟出来是吧？是的。怎么样？是的，这4点20。这个新冠的研发应该说是一个很艰难的一个过程。那我们基本上就是5条、10条线，把串联变成了并联，然后呢，每条线都要就是在设计的过程当中 都要尽可能的不犯错误，少犯错误。再往下呢，能够节约时间，然后呢，在这个大量的实验数据面前，能够找出然后正确的这个结果。
喂，哎，谭主任，什么事情啊？对，明白啊。
我记得应该是1月17、18号的样子，谭主任给我打电话，谭老师就说，你这次这个疫苗你必须要做。
At the beginning of 2020, the CDC began advanced planning for the development of a nucleic acid vaccine and began collaboration with a domestic mRNA vaccine R&D team. Given the urgent time constraints, the chief advantage of this type of vaccine lay in the absence of any need to go through the time-consuming process of isolating and culturing a virus. As long as clarity existed regarding the genome sequence of the viral protein, it could be possible to produce mRNA in a lab that could be packaged into a vaccine.
让他还是报一个计划，就是你这个冻干这块，你冻干这条线，你是需要多少的产能，对吧？我们到时候是定10瓶还是定25的？
With 1,273 amino acids in the spike protein of a SARS-CoV-2 virus and over 10 to the 632nd power mRNA sequences that could be translated into the spike protein, failure to make the optimal choice of an antigen sequence had the potential to dramatically impact on the efficacy of the mRNA vaccine. With the help of the Joint Prevention and Control Mechanism, Li Hengwen received powerful support from the IVDC CDC.
这次新冠疫苗的研发，疾控中心在早期，非常及时的给我们提供了这个抗原序列，并且帮我们设计优化了这个抗原。
After obtaining the antigen sequence, Li Hengwen moved on to the next necessary step in all vaccine production: animal testing. In the small courtyard of the ILAS, which is short for the Institute of Laboratory Animal Sciences, scientific researchers are commemorating the dead. But they are not commemorating the lives of people lost during the pandemic, but rather the animals who gave their lives in the cause of vaccine development. These little animals who made sacrifices for human health are worthy of being appreciated and remembered by us forever. But the sad fact remains that animal testing is still one of the cornerstones of vaccine development. Every vaccine under development must go through screening and evaluation so that the scientists can be sure about the antigen that triggers an immune response. Such tests may last for years.
要去研究人类的疾病，我们要先把这个疾病复制到动物身上。那么这种能够模拟人类疾病的这个动物的模型，我们就叫它疾病动物模型。
Common animal subjects for testing include rats and mice and other animals that reproduce quickly and form stable populations. This makes it easier for them to be sampled in experiments. But there's something else that's needed in order to create animal models for vaccines: live viruses. These were in extremely short supply in labs during the early phase of the COVID-19 pandemic. So, as soon as the IVDC managed to isolate the suitable strains of virus, they began to share them with affiliated research institutes.
我记得那天晚上还是有点小雪的，应该是2020年1月23号，在第一时间把我们这个低度最高的这个病送到了科院的实验动物所。
Thanks to the timely delivery of the strains of virus, the stage was set to begin animal experiments at ILAS.
就大一些，就能给临床提供 更多一些的参考依据，对吧？你在上疫苗的时候，或者是我们在找治疗方法的时候，没有动物模型根本不可能。
The task Qinchuan's team at the ILAS was twofold: to monitor the clinical responses of animals and assess the efficacy and safety of COVID-19 vaccines in animals.
我们观察了它的短期的病变、长期的病变、严重的病变和轻的病变。因为动物它不是人啊，你必须把这个比较分析好了，几组的同时上，最后确定一种方法学，确定一个数据。那这个时候就已经是日以继日的工作了。
In the P3 lab at the ILAS, scientists turned to laboratory animals in their quest for answers. As it happened, Qinchuan's team had achieved a breakthrough of major significance in the SARS-CoV outbreak 17 years earlier, the transgenic mouse. Since the ACE2 gene in mice is different from that of humans, they cannot be directly infected with the coronavirus. The only reliable way to make them into viable subjects for experimentation was to make them more similar to humans. Microscopy was key to this process. Using a glass needle of 0.1 to 0.5 microns for microinjection, researchers directly injected exogenous human genes into mouse embryos to create a generation of rats and mice that could be infected with the coronavirus. It was largely thanks to the groundwork done by Qinchuan's team almost 2 decades earlier that, when the time came to develop the COVID-19 vaccine, the experimental cycle of preclinical animal testing could be shortened from 1 or 2 years to just a few months.
这次的像我们这所做的工作呢，都是世界第一个来做成的这件事情。第一个动物模型就为疫苗的可行性就打了基础。
我们必须在动物身上证明安全有效，然后呢，国家的这个药监局对这些数据进行审核，因为它这个结果可信，再批准他们进行人体试验。
The team, led by Qinchuan, completed the evaluation of 80% of the COVID-19 vaccines deployed by the nation, including the first COVID-19 vaccine entering clinical trial in the world, the first COVID-19 vaccine approved for emergency use, and the first market-ready COVID-19 vaccine. The team originated the technical system of the protective effect evaluation of the COVID-19 vaccine on animal models, and releasing it to the world through the journals Nature and Science. It had been 35 days since Chunwei's team, stationed in Wuhan, then epicenter of the epidemic, had begun their scientific researches. At 4 a.m., February 29th, the first batch of recombinant adenovirus vectored vaccines arrived at Wuhan. Chunwei, developer of the vaccine, as well as 6 scientists from the Academy of Military Medical Sciences, became the first group of subjects for COVID-19 vaccines in China.
我们疫苗是凌晨运到的，12月29号凌晨4点多钟运到。我这是在想，我们能做什么呢？先走一步，早走一步，这些人体数据对我后面的包括临床方案的选择，包括免疫程序的选择，包括让更多的志愿者能够相对有勇气来试我们疫苗，我觉得可能都是一个最好的现身说法。
In fact, Chunwei was surprised at the enthusiasm that greeted the call for volunteers. The announcement of a quota of 108 volunteers met with 5,346 applications in 2 days.
17的时候，武汉还在封城，而且那时大家对新冠病毒了解还不是很深刻，应该说很恐慌，应该说，所以来做一个疫苗的这个受试者。 其实也需要很大的这个勇气的，应该说。
On March 19th, 2020, despite the ongoing lockdown, a group of over 100 Wuhan citizens ventured out. The first phase of the clinical trial of the recombinant adenovirus vectored COVID-19 vaccine developed by Chunwei's team had begun in Wuhan. During the phase 1 trial, subjects had to face the risk of suffering unwanted side effects. Due to differences between individuals and the characteristics of the vaccine, the possibility existed of suffering fever, localized swelling and induration, and even severe allergic shocks.
作为第一例已志愿者的参加疫苗的接种，十分紧张，也很激动。武汉加油，中国加油！
Results were promising. Major cellular immune responses in all 108 volunteers. In Wuhan, the eye of the storm, finally there was a ray of hope. The hope of an effective SARS-CoV-2 vaccine. As the outbreak had been basically contained within China due to highly effective non-medical interventions taken by the Chinese government, the Phase 3 clinical trial was to be conducted in overseas regions where there was high epidemic prevalence. Clinical trials in vaccine development usually involve 3 phases. Phase 1 is aimed at monitoring the safety of the vaccine. It usually involves up to 100 subjects. Moving on to Phase 2, several hundred subjects are divided into 3 groups: a high-dose group, a low-dose group, and another given a placebo. The purpose of this phase is to establish whether the vaccine induces antibodies and to figure out the optimal approach for vaccination. In Phase 3 trials, meanwhile, the number of subjects is expanded gradually. The purpose of this stage is to determine the efficacy and safety of the vaccine. During this stage, it is necessary to create opportunities for subjects to be exposed to the virus. On June 23, 2020, Zhu Jingjin, Vice President at the China National Biotech Group Company Limited, China National Pharmaceutical Group, led the Great Wall Squad to the UAE. This trip marked the official start of the Phase 3 clinical trial of Sinopharm vaccine in the UAE, the first international collaborative clinical trial on an inactivated COVID-19 vaccine in the world.
我们临床试验呢，第一批的人员呢，一共是29个人。我们把这个3期临床试验取名为长城，寓意着是健康的长城。我们来到了阿联酋以后呢，以此为临床试验的中心。
The increasing quantity of samples and data from clinical trials carried out in different regions greatly sped up the process of developing a market-ready COVID-19 vaccine. The number of volunteers was crucial to the final result of the trials.
我自己感觉到的就是压力特别大，有没有人愿意参加这个实验？接种到了5000人的时候，无论是接种者还是被接种者，大家都在鼓掌，哎呀，特别高兴，觉得我们真是有希望了。
By November 2020, the Phase 3 clinical researches of the inactivated COVID-19 vaccine developed by China National Biotech Group, or CNBG, had been conducted in the UAE, Bahrain, Egypt, Jordan, Peru, Argentina and Morocco, etc., involving over 60,000 subjects from 125 countries, including locally based Chinese workers.
得知，我们这个临床入组的人数快要达标的时候，大家都特别激动，都手里面我们原来过11的时候都有那个小的国旗，都拿那个小旗子在唱“我和我的祖国”，非常的激动。
In the meantime, international collaborations were also being carried out. Beginning on July the 21st, 2020, Sinovac successively chose Brazil, Chile, Indonesia and Turkey to carry forward the Phase 3 clinical research of the vaccine.
For this entire vaccine development, without international partnership, we can never make to this level. I think we should thank for all the scientists, all the countries who help us to conduct the trial. And it's not ended yet because we're still keep receiving the real-world study, which is consistent and continued demonstration of the effect of using our vaccines.
But just as clinical trials of the vaccines approached successful completion and preparations were being made to bring the vaccines onto the market, there was a hitch.
你肯定要在28号之前修完。 你恢复生产，你还得2到3天的时间呢。
供应不上，一会儿临没得了。你现在库里还不到1车，不到10吨。
你肯定要去看一眼。
Just as vaccine production was gearing up, there was a shortage of the vials necessary for storing individual doses. This was a major headache for Dai Ji-chu's team. Gaining access to a supply of medical-grade vials made with the non-reactive borosilicate glass needed to guarantee the integrity of the vaccine would not be easy. Data showed that the global stock of medical vials amounted to 200 million, well short of the need to vaccinate a world population of nearly 8 billion. It was a problem that all vaccine manufacturers would have to face up to.
瓶子就意味着什么？你虽然我们国家有疫苗了，但是它很困难，你没有瓶子，你没法包装，没法包装，你就相当于光有疫苗，投入不了市场，这是很麻烦的事情。
Bottleneck was the term that frequently came to Dai's lips to describe the shortage. To him, it was infuriating to think that having done the hard work of developing the COVID-19 vaccine in the shortest period possible time, the battle against the virus might be stymied by something as mundane as a shortage of tiny bottles.
这次任务很重，从董事长一直到我们的彭超院士，都要求我们这个月必须把围沟的预工工给拉出来，时间很短了，所以难度非常大。
Stepping up production of vials was problematic because they had to be made of a stable and low-temperature resistant material, neutral borosilicate glass. Fortunately, measures put in place in 2017 pointed to a solution. Then Dai Ji-chu had helped the company obtain the patent on self-developed neutral borosilicate glass tubes, so they were no longer dependent on imported ones. Now his company is the biggest manufacturer of the product in China. But they weren't out of the woods yet. The need to ensure output sufficient for the vaccine rollout meant that production lines were operating at high capacity. Breakdowns and malfunctions were unavoidable.
每期我们整天都在生产线上观察调整。这个水从哪地方过去的？这个沙子难度很大的，这个是极具挑战性的。所以我们也在制定了方案出来，也在做调试尝试。
After 3 days and nights' heroic efforts supported by expert and eagle-eyed production line troubleshooters, the glass vials were finally ready. As 2020 drew to a close, there was good news for a world long tormented by the scourge of COVID-19. And not just in China. On December the 2nd, 2020, the Pfizer-BioNTech COVID-19 vaccine from the US was approved by the Medicines and Healthcare Products Regulatory Agency in the UK. On December the 31st, 2020, the Joint Prevention and Control Mechanism of the State Council of China announced that the Sinopharm BBIBP inactivated COVID-19 vaccine had received conditional marketing authorization from the National Medical Products Administration. On February the 5th, 2021, the CoronaVac inactivated COVID-19 vaccine, developed by Sinovac, was also given conditional marketing authorization in China.
整个疫情在全球肆虐，只有灭活疫苗是安全有效，就这4个字是确定无疑的。
Beginning in February 2021, the Chinese COVID-19 vaccines were used in Hong Kong and Macau, and later overseas. To date, people in more than 60 countries, including Serbia, the Philippines and Indonesia, have been vaccinated with the Chinese vaccines. On April the 21st, 2021, Chile's Ministry of Health published a real-world study of protective efficacy of the CoronaVac vaccine, based on data gathered across Chile. At the time, this study was the most comprehensive of its kind, involving more samples than any comparable study globally. The results amply demonstrated the efficacy of the CoronaVac vaccine in real-world situations, especially when it came to reducing the numbers of infected cases requiring hospital and ICU support.
周五的时候做的是一个 CPE 的一个观察，然后周一看到了已经就是。
But despite the launch of the vaccines, the sad fact remained that the threat posed by SARS-CoV-2 was as acute as ever. That's why Tan Wen-jie remains at his post, keeping his eye on the ever-changing virus. As 2021 opened, having passed its initial R&D phase and overcome a host of knotty technical problems, the Stemmerna mRNA vaccine went into production and began clinical trials.
给我们拍着胸脯说可以，但是你也不知道它到底怎么样，对吧？对，他只是说他。我现在要求这个临床实验要全力的往前赶，需要钱需要物，我们全部投入。
The battle against SARS-CoV-2 is bound to be a protracted one. Even though the COVID-19 vaccination has been carried out on a huge scale, SARS-CoV-2 is unlikely to give in easily. That's because viruses of this sort are constantly changing and mutating. It's for this reason that one word has become a cause of such concern globally: variant. On December the 14th, 2020, the first SARS-CoV-2 variant, B.1.1.7, was identified in the UK. In May 2021, the WHO decided to name the SARS-CoV-2 variant lineage with Greek letters. Therefore, B.1.1.7 was renamed the Alpha variant. Later, variants labeled Beta and Gamma were identified, proving to be more infectious and having a higher fatality rate. Worse, they seem to challenge the effectiveness of existing vaccines. So, the surveillance system connecting the national government with the local disease control and prevention units, hospitals, and transfer units, and covering multiple levels, had to be reinforced.
如果突变株保护力下降的情况下，那怎么办？如果是现在的在产生了其他的事情的时候，我们做怎么样的储备啊？这新的突变能不能再保护？
To cope with the variants, top scientists including Wu Guizhen, Tan Wen-jie, and Qin Chuan convened a meeting in Beijing to discuss measures to monitor changes in SARS-CoV-2 and adjust vaccine research and development priorities in light of developments.
新型冠状病毒变异监测及功能影响分析这个重要性我想大家都清楚认真的去按照这个要求来去完成。
主要关注的是这个变异对疫苗、抗体药物对体外的有效性的影响的一个评估，然后呢，还有这个变异本身的对这个感染传播的这些影响，就是它的传播力、致病性和还有这个感染力的一些影响。这里头的话，就是我们利用的主要是体外的，就是分子模型和细胞模型。
那下面我们请课题3的我们课题负责人秦川教授来介绍。
随着突变株的这个到来，我们看到了我们这个实验室要对它做一些那个前瞻性的这样一些研究，建立突变株在多种动物模型的这个体内的疫苗的有效性、药物敏感性的这个评估体系。
现有的新冠疫苗对于这个变异株还是有效的。我们即使有效，我们这个变异株也都筛选种子毒株。
我们在做这个监测项目呢，就是对我们的毒株变异的情况进行监测，同时进行分析也研究新的产品。我们在我们的 P3实验室里头我们用注射疫苗以后产生的血清，然后跟新的这样的一个病毒跟它做交叉实验，看看管不管用。
留给科学持续不断的向科学要答案。某一天，某一个地方，可能我们就找到了一个全新的方案，做出了一种可以防感染的这种疫苗。
新冠病毒的这个变异的话，它每天都在发生。对新冠病毒变异的，就是研究， 因为每天都在进行。无论怎么样，我们都要做到有备无患。
我们对冠状病毒的这个认知还不是非常的这个全面和深入，所以我们正在不断的设立项目，加强我们对这个冠状病毒这个科研的投入。
我们现在要做的是呢，说不只是病毒变异，这个跟踪病毒变异，我们应该怎么去重新去思考，构建这个疫苗，给疫苗的专家们提供信息，给这个政府提供信息，这个制定这个防御方案。
疫苗作为公共产品，要有可担负性、可及性，我们的责任更重了，就是不仅要国内，国际上也要去考虑。
On average, people live 40 years longer than they did at the end of the 19th century. Much of this remarkable improvement in human health and well-being is attributable to the progress of medical science and the widespread availability of effective vaccines. The question now is, in the face of the challenges posed by SARS-CoV-2, will vaccines continue to guard us with their shield of immunity?